Welcome to this lecture on eukaryotic gene expression, basics and benefits. The last few classes we have been discussing about delivering genes into mammalian cells as well as directly into humans, and what are the principles behind this, what kind of vectors are being used to deliver genes into both eukaryotic cells in culture as well as directly delivering genes into humans, and what kind of benefits have come out of it, especially in the area of gene therapy, how people are trying to introduce genes, express genes with the aim of curing genetic disorders. We discussed at length what are the plus points and minus points, what are the problems and what are the prospects, and so on and so forth. Today I am going to introduce to you another very interesting and exciting area of research that again came out of research aimed at expressing genes in mammalian cells and mammalian tissues, especially in animal and human tissues. The question we are going to ask today is that if we can introduce genes directly into humans and with the scope of curing genetic disorders, can we also introduce genes directly into humans and express them so that we can induce an immune response against a foreign antigen or a pathogenic protein, and thereby protect such individuals, immunized individuals, against diseases caused by those pathogens. That is, can we use genes directly as vaccines? So this is an area that has come to be known as DNA vaccines or genetic immunization, again a very important area that developed out of a very simple gene delivery and gene expression system. So let us spend some time to understand what is this area of research called as DNA vaccines or genetic immunization, how the story began, and what are the and where this area is heading to. So we have been discussing the eukaryotic expression systems. We discussed the various non-viral vectors, viral vectors that are being used for delivering genes into mammalian cells. And the last lecture we discussed at length how people are attempting to use these non-viral and viral vectors to express genes directly in humans with the aim of curing genetic disorders, what are the limitations of this approach, and so on and so forth, how far we have succeeded. So what I am going to discuss today is about another very interesting and exciting area of research called as DNA vaccines. As I said, the purpose of this lecture is to introduce to you the concept of inducing an immune response against a foreign antigen by directly expressing genes encoding these foreign antigens in the body. Let me first explain to see how this area of research began and how it developed and where we are heading to. So this just slide summarizes the various viral and non-viral vectors as well as other physical methods that people are using to introduce genes into animal tissues as well as cell lines with the aim of enhancing protein production both in cells and culture as well as in humans and animal systems. For example, we have discussed the various viral vectors that are being used, retroviral vectors, adenoviral vectors, adeno-associated virus vectors, and so on and so forth. We also discussed how people are also trying to use a wide variety of non-viral vectors such as mixing with cationic polymers, liposomes, calcium phosphate transfixion, and so on and so forth, and even electroporation, microinjection, and particle bombardment. The whole purpose of using all this kind of vector systems is to enhance the delivery of genes into maximum number of cells either in the culture or in the animal or human tissues, and enhance the expression of the genes encoded by these vectors. Because gene therapy requires that the protein encoded by the gene has to be expressed at clinically or therapeutically significant levels. Only then that you can call this gene therapy as successful. So with the aim of enhancing the efficiency of transfection as well as enhancing the expression of the transgene, a number of these vector systems are being used to have some therapeutic potential. This slide just tells you what are the major advantages and disadvantages of these various vector systems. For example, if you take the viral systems, they are compared to non-viral systems, the viral systems are highly efficient in terms of gene delivery, but in terms of economics they are very expensive. It costs a lot of money to clone your genes to viral vectors and generate a recombinant virus in sufficient titers and use them either in vivo or ex vivo approaches. Whereas in contrast, if you look at the various non-viral vectors, either you complex the gene of interest lipids or you use what is called as a gene gun or a particle gun, where you take your genes or plasmids, coat them with gold particles and directly shoot them into tissues. We will discuss this in a little while from now. The purpose of all this, what I am discussing here, is to see, if you look at the efficacy versus economics, techniques which are very good in terms of efficiency of gene delivery as well as expression of the genes, they are also very expensive. Among all these things, if you try to inject your plasmid DNA, what is also known as a naked DNA, directly into tissue, it is very inefficient. As I said, the cell membrane is negatively charged and acts as a barrier for the entry of the negatively charged DNA inside the cells. Therefore, if you try to just inject directly into your plasmid DNA into cells or tissues, the cells do not take up the DNA very efficiently. So, in terms of efficiency of gene delivery and expression, the plasmid DNA, trying to introduce an expression plasmid directly into cells, the most least efficient of all the vector systems we have discussed so far. But at the same time, it is the most economical method of expressing genes. So, it is for this reason, it is because of this major limitation, that is, the DNA or the plasmid DNA or an expression vector encoding a foreign gene cannot be taken up by mammalian cells very efficiently, that we are trying to put them in viruses, or we are trying to complex them with the lipids and so on and so forth, to see whether we can enhance the efficiency of gene delivery and gene expression. But today I am going to focus on this least efficient or most inefficient method of introducing genes into the cells, and explain to you how this very inefficient but very economical method of gene delivery found a very new application in the form of inducing an immune response for a foreign protein or a foreign antigen, and how this field has now come to be known as genetic immunization or DNA vaccination. Now, what is a naked DNA? A naked DNA is nothing but it is an eukaryotic expression plasmid that is not complex with any chemical formulations, or neither it contains any viral components. So, it is just a eukaryotic expression plasmid. It contains a mammalian promoter for driving the expression of the foreign gene and other sequences which are required for a typical mammalian expression plasmid. The problem of using such an expression plasmid for introducing or for expressing genes in mammalian cells is that it is known for a long time that such plasmid DNA is not taken up efficiently by mammalian cells. As I said, this is the reason why you have to complex this DNA with either lipids or do electroporation or try to do particle bombardment or use viral vectors and so on and so forth. This is the major limitation of using directly DNA, administering cells directly the DNA. But very interestingly, in the year 1990, Wolf and his colleagues demonstrated that skeletal muscle cells of mouse can be transfected with naked DNA. This was the very surprising result they got, and this was actually published in the very prestigious scientific journal Science in the year 1990, entitled Direct Gene Transfer into Mouse Skeletal Muscle in Vivo. Because till then people thought that no tissue can take up just the DNA directly. You need to do all these gimmicks that I have explained so far. Only then some efficiency can be brought into. So, this possibility, this finding that skeletal muscle of mouse can be directly transfected with the DNA, opened up new possibilities for delivering genes in vivo without using any chemical or viral components. So, it created a lot of excitement. Now, let us spend some time to examine how exactly these people came up with this particular novel observation. As I said, people are trying to cure genetic disorders using non-viral approaches, and one of the very intensely investigated areas of research in the area of gene therapy is to see, can you now take these expression plasmids or DNA and complex with the cationic lipids. No, these cationic lipids are positively charged lipids, and you complex this DNA with these positively charged lipids and then add this lipid DNA complex to the cells, so that this DNA can be efficiently taken up by cells and your gene of interest can be efficiently expressed. So, a number of research groups were actually trying a number of cationic lipid formulations to see how we can enhance the expression or efficiency of gene delivery into mammalian cells. I have just listed here various papers how people are trying to see whether, for example, can we cure cystic fibrosis by using this kind of a cationic lipid mediated gene delivery, or can we cure certain kinds of cancers such as human melanoma, or can we introduce into the nasal epithelium of patients suffering from cystic fibrosis and so on and so forth. But these two articles which are listed here in blue color is the work being carried out by a person called Phil Felkner in a company called Vical in United States. They were also trying to develop a number of cationic lipid formations to see which of these lipid formulations, if you mix with the DNA, can be most efficiently taken up by mammalian cells or animal tissues in vivo. so that they can now be taken up for gene therapy and so on and so forth. For example, this is one such lipid formulations they were trying, called as DOTAP, which contains a positively charged head and a hydrophobic tail, and when you take such cationic lipids and then put the DNA, the plasmid DNA gets interacts with this positively charged head group, and the DNA gets entrapped by a lipid particle, and if you now add these cationic lipid DNA complexes into cells, the lipid fuses with the cell membrane and the DNA is delivered inside cells. So, variations of these cationic lipids, a number of cationic lipid formulations were being synthesized by organic chemists, and they were trying to see which of these lipid formulations, if you mix with a DNA preparation, can, and then if you inject such cationic lipid DNA complexes into various tissues, either intravenous route or intramuscular route, which of them will most efficiently deliver your gene or a plasmid DNA efficiently into tissues. When they were doing this, that is, when they were assessing the efficacy of these various cationic lipid formulations for transfecting plasmid DNA into mouse skeletal muscle, they were using actually just the plasmid DNA alone as a control. That means, plasmid DNA mixed with a number of different cationic formulations, for which the plasmid DNA alone served as a negative control. So, what was expected is that the plasmid DNA alone should not get into the muscle tissue very efficiently, but when you mix this plasmid DNA with other lipid formulations, there should be an enhancement of the efficiency of gene delivery into the tissue. Very surprisingly, what they found is that transfection of mouse skeletal muscle was more efficient when naked DNA was used alone rather than complex with cationic lipids. So, this is a very surprising result that they got, which was what published in the journal Science in the year 1990. So, the expected result was that when you take this plasmid DNA and mix with various cationic lipid formulations, one or the other of these lipid formulations would efficiently promote the efficiency of gene delivery into the mouse skeletal muscle. But surprisingly, these people found just injecting plasmid DNA dissolved in saline was being taken up by the mouse skeletal muscle much more efficiently than mixing this DNA with various cationic lipid formulations. This was kind of against the dogma at that time, because DNA cannot be efficiently taken up by mammalian cells. In fact, this is some of the highlights of this research paper which was published by this group. They were actually using a number of genes encoding reporters like chloramphenicol acetyltransferase, luciferase, beta galactorase, because it is very easy to detect the expression of these reporter genes. So, you take eukaryotic expression plasmids coding for either chloramphenicol acetyltransferase, luciferase or beta galactorase, which are all very well known reporter genes, and then you complex them with various cationic lipids and inject them into the mouse skeletal muscle and see which of this tissue will show high levels of beta galactorase activity or chloramphenicol acetyltransferase activity or luciferase activity. So, that lipid can now be taken up and see whether it can be used for gene therapy. That was the whole idea. When we are doing all these things, they found that instead of mixing with all these cationic lipids, if you now take this plasmid encoding for either of these reporter genes and directly just dissolve them in saline, in phosphate buffered saline, and inject them into muscle tissue, you can see in and around the injection site, if you now look for beta galactorase activity, you can see blue colored cells expressing beta galactorase, indicating that cells in and around the site of injection have actually taken up the plasmid DNA, and the gene that has gone inside the cells, and the gene is getting expressed, and you are able to detect the beta galactorase activity. This is just a magnification of one such area. You can actually see of the various muscle cells. Here are a few muscle cells which are blue in color. Of course, this is a black and white picture, but these are actually blue colored cells, indicating that these cells have actually taken up the plasmid DNA which has been designed in saline, and the beta galactorase gene expression can be detected inside these muscle fibers. So, this was a very surprising observation, because none of the tissues are supposed to take up just the plasmid DNA alone. It requires a lot of other gimmicks to get into the mammalian cells. So, this created a lot of excitement, and that is the reason this paper got published in Science, a very prestigious journal. So, people thought, on one hand, people are trying a number of gimmicks using viral vectors, non-viral vectors to introduce genes into mammalian tissues and human tissues to enhance the expression of the transgene, so that you can have applications in the area of gene therapy. And here is a situation where you do not require any of these delivery systems. If you simply take a plasmid DNA, dissolve in saline and simply inject into the skeletal muscle, the muscle cells seems to be able to take up the DNA and express. So, this immediately raised a huge possibility that instead of putting beta galactorase or chloramphenicol acetyltransferase or luciferase genes, can you now put a gene coding for a factor VIII, for example, which is a clotting factor, and in patients suffering from hemophilia, for example, they don't produce factor VIII, and therefore if you now simply take the gene coding for factor VIII, put it in a expression plasmid, and if you simply inject this expression plasmid into muscle, skeletal muscle, will now the factor VIII be made inside the muscle cells and will it be secreted into the bloodstream, so that the factor VIII, the hemophilia patients can be cured of this disease. The reason why this kind of a thing is very attractive, because for as you know, hemophilia is an X-linked disorder affecting 1 in 10,000 males, and this disease is due to a defect in the gene coding for blood clotting factor called factor VIII, and normal circulating levels of factor VIII is about 200 nanograms per ml. So if we are all normal, if we do not have any blood clotting disorders, our blood contains about 200 nanograms per ml of this clotting factor. That is why whenever there is a cut and when we bleed, immediately bleeding stops. But to be therapeutic, patients suffering from hemophilia in which this gene is defective, even if you can get 20 to 40 nanograms per ml, that is 1/10 the actual levels found in normal individuals, it is still considered clinically significant. So the aim of gene therapy researchers who are trying to cure hemophilia is to see, can we develop a gene delivery system to express factor VIII at least to these levels, which is even 1/10 of that found in the normal individuals, so that these people does not have to take factor VIII injections and so on and so forth. So one of the first aims of this kind of a direct gene transfer using naked DNA was to see, if you now take a plasmid DNA and put a gene coding for factor VIII and just inject into the skeletal muscle of these individuals, can you get this much level of factor VIII in the blood circulation? But soon it became very clear that intramuscular injection of plasmid DNA encoding genes like factor VIII gene did not result in the production of clinically significant levels of factor VIII. So although the studies with reporter genes were very exciting, when it actually came to very important therapeutic genes, they found that this technique failed to deliver clinically significant levels of proteins could not be produced using this naked DNA injection. So which tells that it cannot really be used for gene therapy. Similarly, people are trying to say, can you, by simply introducing this kind of a naked DNA, can you cure disease like Duchenne muscular dystrophy? You know, Duchenne muscular dystrophy is a skeletal muscular disorder. The protein called dystrophin is not produced in these individuals, and therefore they suffer from this disease. So again, the idea is to say, can you now introduce the dystrophin gene into the skeletal muscle fibers of these individuals, so the dystrophin is now expressed in these muscle fibers and they can be cured of the disease. But again, when they did the same naked DNA injection, they found that only 1% of the myofibers could be transfected with the plasmid. So indicating that transfer of dystrophin gene into myofibers of Duchenne muscular dystrophy patients could be beneficial, but a larger number of genetically modified myofibers will be necessary for a clinically meaningful result. So both using things like hemophilia or Duchenne muscular dystrophy, it became very clear this technique of directly injecting plasmid DNA into muscle cells to express genes of your interest may not be suitable for the purposes of gene therapy. This is the paper that came out sometime in 1991. The other thing that became very evident is that while the naked DNA injection seems to be reasonably efficient in mouse skeletal muscle, when they did the same thing for higher mammals like primates, the primate muscle fibers were not getting as efficiently transfected as that of mouse skeletal muscle, indicating that there are species specific differences. The efficiency of naked DNA uptake is much more efficient in the mouse skeletal muscle, but is not as efficient in primate muscles. So the efficiency of gene transfer to skeletal muscle in primate muscle fibers is relatively low and variable, indicating that this may not really work in humans. So the initial excitement that naked DNA or just directly injecting plasmid DNA can have applications in the area of gene therapy soon turned out to be a false hope. So because the efficiency of gene delivery is not very efficient, and therefore it cannot be used for gene therapy. So it failed as a gene therapy technique. Then this, as I said, the first report came in the year 1990, and for the next 2 to 3 years, a lot of people tried to see whether they can use this technique for gene therapy, but it became very clear the technique is very inefficient, therefore cannot be used for gene therapy. So the excitement died down. Then this paper came in the year 1993, entitled Heterologous Protection Against Influenza by the Injection of a DNA Encoding Viral Protein. What this group headed by Ulmer et al. Actually demonstrated is that instead of taking genes coding for things like factor VIII or dystrophin, you now take a gene that codes for a foreign antigen of a virus or a pathogen. So, you replace the gene, therapeutic genes like dystrophin or factor VIII, and put a gene that codes for a foreign antigen like a viral protein. And now if you take this plasmid and inject to the skeletal muscle of a mouse, the foreign antigen gets expressed in the skeletal muscle, and it should be presented to the immune system of our body, and this immune system, in conjunction with either class 1 MHC or class 2 MHC, when they express foreign antigen, we should be able to develop either a very potent antibodies or a very potent cytotoxic T lymphocyte response. And this is what this author showed here. So, although this technique of naked DNA injection failed as a gene therapy technique, when they took a gene that goes for an influenza protein, influenza antigen, influenza is a very nasty virus, causes a lot of problems even now, and it is very difficult to develop influenza vaccines. So, if you now take a gene that codes for what is called as a nucleoprotein of an influenza virus, put it in expression plasmid driven by a cytomegalovirus promoter, simply if you inject into the mouse skeletal muscle, they found the influenza protein that is getting expressed inside this animal tissue is able to evoke a protective immune response. This is what was showed. Immunization of mice by intramuscular injection of plasmid DNA encoding an influenza viral protein generated specific cytolytic T cells and protected mice against subsequent challenge of a live influenza virus. This is a very novel concept, because if you want to induce an immune response against a foreign antigen, you have to take this foreign antigen, you have to produce, like we discussed in the earlier classes, like hepatitis B antigen for example. If you want to make a hepatitis B vaccine, you have to take the gene coding for hepatitis B virus, express in yeast cells, purify this protein, mix with adjuvants such as alum, and then only if you inject, then only you will get an immune response. But here we are saying that you do not have to do any of these things. Simply take the gene coding for a foreign antigen, clone it into a mammalian expression plasmid, make this plasmid in large amounts in bacterial cells, and simply inject the plasmid DNA into the mammal, in this case mouse, and this antigen is now made inside the muscle cells and is now able to induce an immune response, which means you do not have to express a foreign protein or you do not have to purify a foreign protein. Direct injection of a gene coding of a foreign antigen can induce an immune response in a mammalian system, which is a very novel observation in the early 1990s. This generated a lot of interest. So, number of reviews and news and views came out in the journal saying that naked DNA points to vaccines. So far the dogma has been that only when you immunize animals with proteins or carbohydrate conjugated to proteins, you can induce an immune response. But for the first time, these people have demonstrated that you do not need a protein or a carbohydrate based immunization. Genes can be directly injected into the body, and these genes, when injected into the body, the proteins will be made in situ inside the cells of mammal, and it can induce an immune response. So, genes can be used as vaccines, which created a lot of excitement. This is the actual experiment that was done by this group, which was published in this paper. They took these mice and injected them with a plasmid DNA that codes for what is called a nucleoprotein of the influenza virus, and then after giving 2 or 3 immunizations, challenge these mice with the actual influenza virus. And as you can see here, if you look at the % survival versus days after viral challenge, as after 5, 10, 15, 20, 25 days, by 15 days, all the control mice which were not expressing the antigen, they all died because of the virus challenge, but all those mice which were actually expressing the nuclear protein or which were immunized with the nuclear protein expressing plasmid, they were all protected. The other important point in the experiment is that the challenge virus that was used is called a heterologous challenge, because the nucleoprotease gene came from a different strain of a virus, and the challenge virus was another strain, which was unheard of in the case of influenza, because influenza virus keep changing its surface epitopes very often, and therefore a vaccine which was made, for example in the previous year, will not work against the strains which are in the field the following year. So every year vaccinologists or vaccine manufacturers have to go to the field, see what kind of influenza virus are circulating in this field that particular year, and then use these virus strains for making new vaccine. So every year you have to go to the field, pick up the circulating viruses, and then make them as vaccine, develop them as vaccine strains. So this is a major problem in the influenza. But here you have a situation, this is actually shown in this clear. The antigen gene coding for the nuclear protein of the influenza virus came from a virus strain called H1N1, which was isolated in the year 1934 from patients. But the virus which was challenged, the challenge virus, was a virus strain that created an epidemic in the year 1968. So, the vaccine was made from a different strain of influenza virus. The challenge virus is a different strain, but still it conferred protection, indicating that there was a cross strain protection using the DNA vaccination. Now, I have just described in detail some little bit of immunology behind this. Viruses escape immune responses by mutating the regions against which an immune response is generated. This is what I told. Usually, when you take influenza virus, an inactivated influenza virus, and give it as a vaccine, all the antibodies will be generated against what is called as the hemagglutinin protein, which is present on the surface of the virus. But since these viruses keep changing this hemagglutinin protein now and then, if you make a vaccine against one particular strain, and strains which have changed the hemagglutinin a few seasons later will no longer respond to the vaccine. The vaccine will not be able to protect such kind of a strains. That is why you need to keep changing your vaccine strain as often as possible. So, antigens which are expressed on the viral surface are often the targets of generation of neutralizing antibodies, and when these surface antigens are mutated, antibodies can no longer neutralize the virus. This is the problem with viruses like influenza or even malarial parasites or even HIV and so on and so forth, because they keep changing the surface antigens. On the other hand, viral proteins which are internal are often ineffective in inducing antibody responses, but they can induce very potent cytolytic T cell responses. In order for an antigen to induce an antibody response, they should be present on the surface of the virus. Only then the antigen presenting cells can see them and then induce an antibody response. But for inducing a cell mediated immune response, the antigens have to be synthesized inside the host cell, and these antigens has to be presented in complex with class 1 MHC on the surface of the infected cell. In this case, on the surface of the transfected cell, and then this will be recognized by these CTLs, and you get cytotoxic T cell response can be induced. So, in order for the internal antigens to induce a cytotoxic T lymphocyte response, they need to be synthesized inside the host cell and be present on the infected surface by class 1 MHC. These internal proteins, so unlike the surface proteins which are present on the viral surface, these internal proteins are highly conserved. They do not change that often as the surface proteins do. So, the internal proteins are often conserved between various clades or various strains of this virus. So, this is the reason these people actually use the nuclear protein of a different strain, and when the cytolytic T cell response was generated by this nuclear protein, he is able to confer protection against a totally different strain which came out almost 13 years later, because the nuclear protein is pretty conserved in these strains. So, DNA vaccination led to protection against a strain of an influenza virus that not only was a different subtype from the strain from which the gene of the DNA vaccine was derived, but it has arisen 34 years later. So, the nuclear protein gene came from a strain which was in the year 1934, the challenge virus came out in the year 1968. So, even after 30 years, this nuclear protein gene is able to confer cross strain protection. So, it was a very major breakthrough. So, this created a lot of excitement among vaccinologists, indicating that almost all major newspapers reported this finding as a very exciting delivery, and in fact, this was being called as a third vaccine revolution in the area of vaccinology. And this year 1993 actually marked the beginning of this DNA vaccine research. The reason why I am emphasizing is that you can see here a very simple gene delivery technique. You simply take a gene coding for a viral protein, put it in a mammalian expression vector, just inject them into skeletal muscle, and the antigen is made inside the cells and the antigen is expressed, and you are able to induce an immune response. So, a simple gene delivery technique which failed as a gene therapy technique became a very promising technique for inducing an immune response and created a whole field of research called DNA vaccination or genetic immunization. What are the advantages? Why people are excited about these DNA vaccines? They have a lot of advantages compared to the conventional vaccines, which is either live attenuated vaccines or inactivated vaccines or recombinant protein based vaccines or carbohydrate vaccines, because they are very simple and easy to produce. Cloning a gene into a eukaryotic expression plasmid and making this plasmid in large amounts E. coli is much more less expensive and much more simpler than infecting cells with viruses, killing the viruses, or developing live attenuated strains of viruses, or making recombinant proteins, purifying these proteins and giving it as vaccines. So, producing a plasmid DNA is much cheaper and much simpler than producing other kinds of vaccines. The other important advantage of DNA vaccine is that it does not require a cold chain. All the other vaccines that I talked so far has to be refrigerated. whereas DNA is much more stable than proteins or inactivated or live attenuated viruses. So, plasmid DNA need not be stored in the cold chain or refrigerator. So, it can be stored at room temperature. In fact, almost 20 to 25% of the vaccine cost often goes for maintaining what is called a cold chain. That is, from the place of manufacture to the ultimate place of use, you have to keep this vaccine in refrigerator conditions. So, if we can develop a vaccine which does not require this cold chain, the cost of vaccine can be dramatically brought out. So, there was a lot of excitement when people showed that DNA can be directly used as a vaccine, that suggested that you do not have to store them in refrigerators. So, the cost of vaccines can be dramatically brought down. It is non-infectious, there is no risk of infection because you are not dealing with pathogens. Remember, if you have to make a live attenuated vaccine or if you have to make a killed vaccine, you have to infect cells with the virus, grow this virus in large amounts, either use these attenuated viruses or killed viruses as vaccines. So, that means you have to deal with the pathogens. Here you are only dealing with 1 or 2 antigens of the pathogen. So, it is non-infectious and it is very versatile because it can activate both cellular as well as humoral arm of the immune response. You can get a cell mediated immune response, cytotoxic T cells, you can also get very efficient antibodies. And the other advantage is that there is a lot of flexibility. You can stitch whatever genes you want, you can put different epitopes, you can make synthetic genes containing different epitopes from different antigens and express them so that you can develop polyvalent vaccines. So, because of these numerous advantages, DNA vaccines generated a lot of excitement. Similarly, the other advantages are, compared with other types of vaccine technologies, DNA vaccines have much more widespread applications. For example, once the manufacturing and purification process for a plasmid DNA is established, a similar process can be applied for other different vaccines. Only difference will be that for in one case, for example, if you say if you want to make an influenza vaccine, you would be putting an influenza gene, a gene coding for an influenza protein. Whereas, if you want to develop a vaccine for a malaria, for example, instead of an influenza gene, you will be putting a gene coding for a malarial antigen, but otherwise the rest of the vector is the same. So, in terms of manufacturing plasmid DNA, whether it contains an influenza gene or a malarial antigen does not make any difference. So, the manufacturing process will be more or less the same, only the inserts will be different, the genes will be different. So, that means you can, using the same manufacturing facility, you can produce different DNA vaccines, which is a tremendous advantage. Whereas, if you have to make attenuated or inactivated or recombinant protein vaccines, each process is different for a different vaccine. A process which is used for making a recombinant hepatitis B vaccine cannot be used for making a polio vaccine, because this manufacturing process is totally different. So, all these advantages made the entire area of DNA vaccine research very exciting. So, a lot of interest was generated to see this simple gene delivery and gene expression technology, can it be developed as a very novel form of inducing an immune response in mammalian systems. Once people demonstrated that just by injecting DNA, plasmid DNA encoding foreign antigens can induce an immune response in skeletal muscle, people ask the question, why skeletal muscle? Can you give it to other tissues? In fact, instead of injecting plasmid DNA into skeletal muscles, if you now take this plasmid DNA encoding foreign antigen and mix it with gold particles and then shoot them, propel them into the skin by what are called as gene guns, then they found the efficiency of immune response is much more. You can get a more efficient immune response if you introduce these foreign genes or foreign expression plasmid encoding foreign genes into skin using biolistic methods than simply injecting using syringes into skeletal muscle. So, new devices called as gene guns were actually developed. For example, a paper that came in Nature in the year 1992 entitled Genetic Immunization, a very simple method of eliciting immune response, suggested that for inducing the dogma till then, for example, to produce an immune reaction against a foreign protein, you you need to have a purified protein of that particular organism. Then only you can, if you inject that in presence of adjuvants, you can get an immune response. But the isolation and purification of proteins is time consuming and sometimes very difficult. If these antigens are produced in very small amounts, or if you cannot express them properly, then you cannot make a vaccine for that particular antigen. Immune response can be elicited by introducing gene encoding protein directly into the skin using a handheld form of a bioelectric system which can propel DNA coated gold particles direct into the cells. This is the picture of what is called as a gene gun marketed by a company called Biorad. So, instead of making the protein, purifying the protein and then mixing them with adjuvants and injecting to induce immune response, here is a very simple method. You simply take the gene, put it in a mammalian expression plasmid and then mix it with certain gold particles, and using this kind of a gene gulls you can just shoot them into skin and you get a fantastic immune response against this foreign antigen. So, this is the enlarged picture of this gene gun called as the Helios gene gun, which was marketed by Biorad. So, you can simply take your DNA containing your foreign antigen gene, coat them with gold particles and simply put it into this device and just shoot them into your skin, and the DNA will be going to the skin and the antigen will be made inside the skin cells and you will get an immune response. So, we will spend a couple of minutes to understand how exactly this DNA vaccine is able to induce an immune response. What is the mechanism by which injecting this plasmids either into skeletal muscle or shooting them into skin is able to elicit an immune response. So, how does injection of plasmid expressing a foreign antigen into skeletal muscle induce an immune response? Now, in order to elicit an immune response, antigenic peptides need to be expressed on the host cell surface in conjunction with class 1 or class 2 MHC. I am sure all students who have studied basic immunology knows this. If you want a cytotoxic T lymphocyte response, the antigens has to be synthesized inside the cells through the intracellular pathway and should be expressed along with class 1 MHC, and then only you will get a cytotoxic T lymphocyte response. Whereas if you want an efficient antibody response, it has to be expressed in conjunction with class 2 MHC and then you get a very efficient antibody response. So, class 1 molecules, class 1 MHC molecules are expressed by almost all nucleated cells. However, the expression of these class 1 MHC molecules is very high in antigen presenting cells such as dendritic cells, Langerhans cells, etc. Whereas other somatic cells such as fibroblasts, liver cells, muscle cells do not express very high levels of class 1 MHC antigens. What it tells you is that if you introduce your DNA into somatic cells such as skeletal muscle cells, because the muscle cells do not express class 1 MHC at very high levels, the antigen presentation will not be very efficient. Whereas if the same plasmid DNA is taken up by professional antigen presenting cells such as Langerhans cells or dendritic cells, since they express very high levels of MHC class 1, the antigen presentation will be much more efficient. So, only antigen presenting cells can efficiently prime cytolytic T cells, only they can induce a potent cytotoxic T lymphocyte response. So, if a non-antigen presenting cell such as muscle cell takes up DNA vaccine and produces the foreign protein or protein antigen, it must deliver this antigen in some form to a professional antigen presenting cells by a process known as cross priming in order for cytotoxic T cells to be induced. So, what is happening when you inject your plasmid DNA? Initially people thought it is the muscle cell which is expressing the foreign antigen and is presenting the antigen in conjunction with MHC, class 1 MHC. But when people realize that muscle is not a very good antigen presenting tissue, people then realize that what is happening when you inject a foreign, when you make this injection containing foreign DNA, you are actually attracting some of these antigen presenting cells to the site of injection, and it is these antigen presenting cells which are taking this plasmid DNA, and the foreign antigen is getting expressed inside these antigen presenting cells, and these antigen presenting cells now express this foreign antigen in conjunction with class 1 MHC, and that is why you are getting a very potent cytotoxic T lymphocyte response. So, what is the mechanism of DNA vaccination? The antigen that is secreted from the DNA inoculated cells is being taken up by the antigen presenting cells, or the antigen presenting cells are directly getting transfected with the plasmid DNA, because they can directly take up the plasmid DNA, because they can by phagocytosis or any other mechanism, and when they present in association with class 1 or class 2 MHC, then you get a very potent immune response. So, it is not directly the muscle cells that is actually responsible for this potent immune response, but it is the cross priming. It is the expression of this foreign antigen by the antigen presenting cells that is responsible for this potent immune response generated by DNA vaccination. The why, when you inject this DNA into skin, you are getting a much better immune response than injecting into muscle cell. This is because skin is a much better immunosurveillant organ compared to skeletal muscle, because number of antigen presenting cells like Langerhans cells, dendritic cells are present beneath the skin, because that is the first line of our defense. Therefore, when you shoot this DNA into skin, or when you do an intradermal immunization rather than an intramuscular immunization, the chances of your plasmid DNA being taken up by these antigen presenting cells, or the chances of the foreign antigen that is secreted from the skin cell and being taken up by these antigen presenting cells are much higher. So, you get much better antigen presentation when you inject your foreign naked DNA into skin rather than into skeletal muscle. So, the Langerhans cell population along with monocytes, granulocytes and natural killer cells comprise the innate arm of the immune response that are either recruited to the skin or recirculated between the skin and blood system in response to infection or trauma. Even by injecting, you are actually creating a trauma. So, in the presence of an infection or a skin vaccination, the innate immune cells interact with the adaptive arm of immune system, the B and T cells that also recirculate to generate a specific immune response to foreign antigen. So, the gene gun delivery system is specifically designed to deliver DNA encoding antigens of pathogen organisms to the immune competent Langerhans cells. So, that is why the intradermal immunization or intradermal delivery of genes was found to be much more better than the intramuscular immunization as far as genetic immunization of DNA vaccination was concerned. So, the first paper on DNA vaccine appeared in the year 1993. Since then, as you can see in this slide, the number of papers were published to actually demonstrate whether it is a viral disease or a bacterial disease, parasitic disease or even for cancer. If you take the corresponding gene or the relevant gene, put it in expression plasmid and either inject into skin or into muscle, you can evoke a protective immune response in animal models. This was demonstrated in the next 5 years following 1993 paper. In fact, more than 42 phase 1 and phase 2 clinical trials in humans were initiated because of this euphoria, because all these plasmid DNA vaccination seems to elicit a protective immune response in animal models such as mice and monkeys and so on and so forth. So, people ask the question, will this magic work in humans? So, human trials were initiated to see, will DNA vaccine work in human beings? A new website called dnavaccine.com was initiated, and even today if you go to the website, you can get updated with all the excitement that is going on in the area of DNA vaccination. People started asking the question, are DNA vaccines safe? A very systematic study was conducted by this group and was published in the journal Human Gene Therapy in the year 1999 to see how safe is this plasmid DNA immunization. And people have actually shown, I will not go into the details of the paper, that following the plasmid DNA immunization in mice, there is no evidence for autoimmune mediated pathology, antinuclear antibodies or antibodies to double strand DNA were observed in the immunized animals, indicating that the plasmid DNA immunization is pretty safe. The other major concern that the DNA vaccination had was, when you inject this plasmid DNA into muscle cell or skin cells, will this plasmid DNA go and integrate into the genome? Remember, in the last class when we were discussing about retroviral mediated gene transfer, one of the major problems of retroviral gene transfer is the random integration of the virus into the chromosome, resulting in the activation of certain oncogenes like LMO2 and so on and so forth, leading to leukemia. No, that was one of the major drawbacks of retroviral mediated gene transfer in the case of gene therapy. In the same way, when you now take the plasmid DNA and inject into muscle cells, people ask the question, if this plasmid DNA now goes and injects randomly into the chromosome of these muscle cells or skin cells, can it activate an oncogene, or can it inactivate a tumor suppressor gene, and as a result can it have very important harmful effects? And a detailed study was again carried out and reported in Human Gene Therapy in 1999. The crux of the study, we can read this abstract of this paper, but what it was shown is that the chances of this plasmid DNA going and integrating into the chromosomal DNA is 3000 times less than a spontaneous rate of integration, chance of spontaneous integration, indicating that the chance that injected plasmid DNA goes and integrates and as a result causes a disorder or disease is 3000 times less likely to happen than that happens in a very spontaneously, indicating that the chances of genomic integration are very, very minimal. The United States Food and Drug Administration, which is the watchdog for giving regulatory approvals for any new drug or a vaccine that comes into the market, actually asked the Centers for Biologic Evaluation and Research to actually bring out guidelines. So, if somebody wants to develop a DNA vaccine for a particular disease, how should they proceed? What kind of qualities of plasmid DNA they have to make? What is the manufacturing practice? What is the contaminant that should be avoided in the plasmid DNA preparation and so on and so forth. So, this agency came out with a document called Points to Consider for Plasmid DNA Vaccines for Preventive Infectious Disease Indications in the year 1996. All these things tells you the seriousness with which researchers as well as companies came in to see whether we can take this new and exciting development of DNA vaccination and make it into a reality. In fact, just 3 years back in November 2007, the Center for Biologic Evaluation and Research of the Food and Drug Administration of the United States came up with another new sets of guidance for the industry, to entitled Considerations for Plasmid DNA Vaccines for Infectious Disease Indications. They revised the guidelines and came up, this is the way you have to make plasmid DNA if you want to inject into human beings and bring this DNA vaccine as a product into the market. Many exciting things happened. Companies actually started using all these guidelines and see can they actually make a clinical grade plasmid DNA. So, if for example, if I am interested in making a DNA vaccine for malaria or DNA vaccine for hepatitis B, I clone this antigen into plasmid, go to these companies and say, can you please make, using the guidelines given by US FDA or the Center for Biological Evaluation, can you use what is called as a good manufacturing practices and make plasmid DNA in a pure form so that they can be used for human clinical trials. So, a new industry came up down to see how we can manufacture plasmid DNA in large amounts, because you require grams and milligram amounts of DNA if you have to inject into humans. So, the entire plasmid DNA manufacture has to be scaled up. So, huge novel methods of plasmid DNA purifications were developed to see how we can purify plasmid DNA in large scale using fermenters and using novel downstream processes. So, a new industry came up following this very simple gene delivery technique. I am not going to the details. A number of guidelines came up to see what kind of manufacturing practices need to be made if we have to make these DNA vaccines for human use, what kind of safety precautions one has to take if we have to examine these DNA vaccines in humans, what are the parameters that you have to check if you have to convince the regulatory authorities that this DNA vaccine is actually safe. I will not again go into the details. All guidelines were actually formulated, and these are all taken from these documents which were actually prepared by the Center for Biological Evaluation and Research CBER. So, other interesting thing that happened following this discovery of DNA vaccination or genetic immunization is that people thought, companies like Genovac came up with this idea that we will make, if suppose we want to make an antibody for a particular gene or particular protein, you do not have to go and then express this protein, clone this gene into a bacterial vector or purify the protein, mix it with an adjuvant and then immunize animals to raise antibodies. They said we will use the genetic immunization procedure and make antibodies and give it to you. This created a lot of excitement, rapid antibody production using genetic immunization. That means you do not have to express these proteins in bacteria, you do not have to mix them as adjuvants and then you do not have to raise antibodies. You can directly inject and immunize animals with these genes and you can make antibodies, which had tremendous scope for basic research, especially in this era of human genome being sequenced and a number of new genes being discovered. We still do not know the function of many of these genes. So, one of the first thing that if you want to understand the function of genes is that you need to make antibodies against these proteins and see where are these proteins getting expressed, what kind of proteins this protein is interacting with. For all these things, antibodies are very important tool to understand the function of novel genes. So, using this approach, what it told you is that if you have a new gene and if you want to make an antibody for a protein coded for a gene, you do not have to take this gene, express this gene into organisms like bacteria, purify the protein and then use it for eliciting immune response. You can simply take the gene, directly put in a eukaryotic expression plasmid, immunize mice or rabbits and you can generate antibodies. So, a new company called Genovac came up with a number of advantages to see how genetic immunization can be directly used for rapid antibody production. It gave just a generalized scheme. One can go to the website of the company to get more details how you can directly introduce antibody production by genetic immunization using mice or rabbits. The company actually demonstrated that you can actually develop very high affinity antibodies using the DNA immunization procedure, and these antibodies generated by the genetic immunization are ideally suited for diagnostic and therapeutic applications. The other important advantage of this kind of a genetic immunization approach is that you can make tailor-made antibodies. For example, if you want only a small antibodies made against a very small region of a protein, you can just delete that, you can clone only that particular region of the gene and express put in an expression vector and you will get antibodies only against that particular epitopes. Or you can stitch different epitopes of a protein, put them together and see whether you can develop antibodies only against those epitopes. Or using genetic engineering and cloning techniques, you can do all kinds of manipulations, genetic manipulations, and you can get tailor-made antibodies for any number of epitopes that you want. You can stitch various epitopes together and put those genes encoding epitopes together and you can generate antibodies for these multi-epitope based vaccines. So, tremendous excitement was there by this discovery of this DNA vaccination. In fact, there's a very nice review that actually says genetic immunization for antibody generation in research animals by intravenous delivery of plasmid DNA that came up in the journal called Biotechnics in February 2006, and one can read this review to understand the scope of using this kind of a genetic immunization approach for inducing an immune response and generating antibodies. Again, people came up with variations of this basic technique. Instead of injecting DNA into skin or injecting DNA into muscle, why can't you take the DNA over the oral route? So, two papers I am just quoting here as example, published in 1998-99, oral gene delivery of chitosan DNA nanoparticle generates immunological protection in a murine model against peanut allergy, and another case for a rotavirus. Protective immunity induced by oral immunization of rotavirus DNA vaccine encapsulated in microparticles. So, you do not need biolistics, you do not need gene gun immunization, you do not have to inject plasmid DNA. If you simply swallow DNA complex with certain chitosans or microparticles, it will be taken through the oral route and you can get an immune response. So, oral immunization using plasmid DNA encoding foreign antigens is possible. Another very interesting paper came in 1999 saying that actually DNA is actually taken through the pores in the skin. If you have mice which does not have the hair follicles, they said you do not get an immune response, but if you have mice which have hair follicles, you get a much better immune response, indicating that when you immunize through the intradermal route, the DNA is actually taken up through the hair follicles and you can get a robust immune response, indicating that this route of DNA delivery is through the hair follicles of the skin rather than the other routes. I think I will not go to the details. Therapeutic DNA vaccines. People thought DNA vaccination in combination with chemotherapy can be used for treating diseases like tuberculosis. Again, a proof of principle paper was published in Nature in 1999, DNA vaccination as an adjunct to chemotherapy. Again, I will not go to the details, but what this tells you is that the excitement that was generated by this very simple and versatile technique. Simply take a gene, put in a eukaryotic expression plasmid, dissolve in saline. If you now simply inject, you can get the antigen can be expressed even though at low levels, but it is good enough to induce a protective immune response, has generated a tremendous excitement between 1993 and till date. Tremendous amount of research is going on in this area of research. I will not go to the details. Again, Stephen Johnson came up with this concept, what is called as expression library immunization, where for example, if I want to make a DNA vaccine for malaria, now I do not know what are the protective antigens. What he says is that simply make cDNA library in a mammalian expression vector and simply inject the cDNA library into the mice, or make batches, take 10 different cDNA clones and immunize and then see which one of this cDNA, then if you challenge these mice and then see which one of this cDNA when expressed can induce a protective immune response. So, by this you can identify novel protective antigens for complex diseases like malaria, HIV and tuberculosis called as expression library immunization. People again came up with a very interesting finding that the CpG sequences which are present in the bacterial vector can actually act as immunostimulatory motifs, because these CpG sequences in the bacterial DNA are not methylated, whereas the mammalian CpG sequences are methylated. So, when you inject a plasmid DNA containing non-methylated CpG, these non-methylated CpG sequences can actually act as immunostimulatory motifs. Again initiated a new area of research in the area of adjuvants. People thought these immunostimulatory motifs or the CpG sequences can actually be as effective as Friend's complete adjuvant or alum. So, a new area of adjuvant research was initiated, indicating that these immunostimulatory sequences, DNA sequences can be used as adjuvants. Our own lab at Indian Institute of Science demonstrated in principle that you can actually develop DNA vaccines for Japanese encephalitis. I will not go into the details. The results were published in Vaccine. We also tried to develop a DNA vaccine for rabies in collaboration with a company called Indian Immunologicals Limited in Hyderabad in India, and in fact regulatory procedures are currently on, and hopefully a product will come out sometime in the near future for a new form of a DNA based rabies vaccine for veterinary use entitled as DNARAB. So, an area of research which began in the year 1993, the first paper on DNA vaccination was published in 1993. It created a lot of euphoria and lot of optimism, but the question comes, in addition to all these publications and all this euphoria, did any DNA vaccines really came out into the market? As I speak today, at least 2 DNA vaccines have been licensed, but both are for veterinary use. One DNA vaccine has been licensed for use in horses for protection against West Nile virus. Another DNA vaccine has been licensed for protection against salmon fish. So, this is the DNA vaccine, West Nile DNA vaccine called as West Nile Innovative DNA, was brought out by a company called 4Dodge, a division of Wyeth, and another DNA vaccine called as Apex IHN, a Canadian company, brought out this vaccine against a viral disease against a fish. So at least 2 DNA vaccines are currently in the market, and hopefully our vaccine will be the third if we get the regulatory approvals for bringing into the market. A number of companies are now trying to use this principle of DNA vaccinations to see can we develop vaccines for diseases like cervical cancer, influenza, HIV, hepatitis C, cancer, and so on and so forth. I will not go into the details. A company called Inovio, for example, wants to use this DNA based technology to see where you can develop vaccines for all these diseases. One can go to the website and look into it, get more details. A company called PowderJet came up with very small devices to see can you inject yourself, can you have DNA vaccine cartridges, you can immunize yourself. Tremendous excitement was initiated. So the purpose of this lecture, what I tried to tell you in the last 1 hour, is to see how a very simple DNA delivery technique which failed as a gene therapy technique but found a new innovation and found a new use as a novel form of inducing immune response in animals and humans. So a technique which failed as a gene therapy technique found very new applications in the form of DNA vaccination or genetic immunization, and how in the last 17 years from 1993 till date, how a whole new area of research was developed based on this very simple gene delivery and gene expression technique, and at least 2 products are currently in the market, both for veterinary diseases, and hopefully some DNA vaccines for human use will soon be approved in the years to come. So I have listed a number of review articles one can actually go through and then enlighten yourself more about the DNA vaccines. Very nice review articles are there, and one can read up many of these review articles and see how people are trying to develop DNA vaccines. In addition, if you go and visit websites like the DNAvaccine.com, you can get more and more information of how this very simple versatile technology of gene delivery and gene expression has tremendous promise of developing into a novel form of vaccination for animals and humans. Thank you.